Hello, everyone, and thank you for joining us today for our webinar, LBA versus LCMS, Competing Technologies at a Development Crossroad. My name is Kimberly Milazzo. I'm the VP of Operations at O'Leary's Colorado Springs site, and I'll be your moderator for this session. We're excited to have you with us as we explore the benefits and considerations of LBA versus LCMS and when to use each tool. The Colorado Springs site has been in operation for 24 years, and much of the exciting technology and research we'll be highlighting today was developed here with our amazing technical team. Before we begin, I'd like to cover a few housekeeping items. First, this webinar is being recorded, and a link to the recording will be available on demand after the session. Second, if you have any questions during the presentation, please type them into the Q&A box, and we'll address them during the Q&A segment at the end of the webinar. We'll be addressing as many questions as possible, and we will answer any outstanding questions via email after the webinar. I'd now like to introduce our speakers for today's session, Shane Karnik and Matthew Hartle. Shane Karnik is a Senior Laboratory Director at O'Leary, where he leads a team of bioanalytical scientists working on projects to support pharmaceutical drug development for both non-GLP and GLP preclinical and clinical studies. Shane received his Master's of Science in Chemistry from the University of Colorado and has extensive experience in the pharmaceutical industry, where he developed and validated analytical methods for new drug submissions. Dr. Matt Hartle is a Senior Analytical Chemist at O'Leary, and he specializes in the application of protein isolation and digestion using innovative techniques like HPLC, MS. Matt received his PhD in chemistry from the University of Oregon in 2016, where he was part of the Pluth research group studying the binding of H2S to biologically relevant metal and non-covalent scaffolds. Without further ado, let's get started. Please join me in welcoming our first speaker, Shane Karnik. Shane, the floor is yours.
Great. Thank you, Kimberly, for that introduction. So today I'm going to be talking about biologics and where they are today in the regulatory landscape. So for the last decade, a growing number of biotherapeutics have been approved by the FDA. For instance, in 2023, of the 55 new drug applications approved by the FDA, more than 30% of the applications were for protein-based therapies, and this number of 30% was down from the previous years. With the rise of protein-based therapeutics, the demand for bioanalytical methods capable of delivering quantitative and meaningful data on these biotherapeutics is escalating. Not unlike traditional small molecule drug development, the journey for biotherapeutics through the drug development pathway is not without its unique challenges. These challenges, coupled with the specific requirements for assaying these drugs, can make the pathway more difficult and time-consuming. For protein-based therapies, traditional LBA assays required the production of high-quality reagents for both capture and detection. The time required to prepare these critical reagents can delay the start of critical decision-making steps. For instance, the time required to prepare antibodies for these assays can exceed 12 months if both capture and detection antibodies are needed. Today, Matt and I would like to discuss the advancements in the analysis of protein-based therapeutics that can shorten the time required to generate IND submission data. Many of these advancements come from the adoption of mass spec analysis early in the drug development process. The types of bioanalysis used for protein-based therapies are predominantly LBA, LCMS, and hybrid LCMS methodologies. Each type of methodology has different advantages. Traditionally, LBA has been the choice for quantitating large molecules, and LCMS has been predominantly used for small molecule drug quantitation. However, recent advancements in mass spectrometry instrumentation has made LCMS a complementary technology for large molecule drug development. There is a growing move towards coupling the advantages of LBA-type cleanup and enrichment with mass spec detection to improve large molecule quantitation. This hybrid approach utilizes commercially available antibodies for drug capture, cleanup, and enrichment. This step is then followed with protein digestion, and the protein is then measured by measuring a surrogate peptide by LCMS analysis for quantitation. This hybrid approach has allowed sensitivity levels to reach or surpass other traditional approaches. Regardless of the methodology used, it is essential to employ well-characterized, fully validated bioanalytical methods to ensure reliable results. This will allow for critical go, no-go decisions along the drug development pathway. Traditional LBA assays are used to quantitate large molecules like antibodies, DNA, RNA, biosimilar, and fusion proteins. For LBA analysis, ELISA and MSD are most commonly used in PK evaluations. These methodologies will require one or two critical reagents to capture and detect the biologic. When using LBA for analysis, the quantitation range is typically 2 to 2.5 orders of magnitude, and the sensitivity level is sub-nanogram per mil. For validation, many of the same experiments required for small molecule drugs are performed, along with additional experiments like minimum required dilution. For LBA methods, additional validation work is often needed when critical reagents are changed during the drug development pathway. This slide shows the typical timing for developing, validating, and performing sample analysis with an LBA methodology. Creating high-quality reagents is a time-consuming step during the pathway and can lead to delay of critical go, no-go decisions along the drug development pathway. At this time, I would like to hand the presentation over to Matt so he can explain how LCMS can be used in the development of protein-based therapies.
Thank you, Shane, for that introduction. LCMS, unlike LBA, quantifies either the entire protein or a signature piece of the protein. This can be divided quite simply into two general processes. Either you can do top-down quantification, which is quantification of the protein itself, where you take your matrix, your cells, your tissues, your plasma, you extract the protein either using gel or your LC, and then you conduct mass spec on the entire protein. This can be challenging from a mass spectrometry side because a typical triple quad instrument has a mass range of about 5 to 2,000 mass to charge ratio, which means that your typical protein is going to need a large number of charges on it in order to be quantified by mass spec. The alternative is to use a bottom-up quantification. This quantification of the protein uses a signature surrogate peptide, and instead of quantifying the entire protein, once the protein is isolated, we digest it into these peptides, which can then be separated by LC to produce characteristic mass spec fragmentation patterns that are then quantitated for data analysis. Importantly, the quantification uses a piece of the actual protein, specifically the surrogate signature peptide of the analyte or protein of interest. You gain selectivity in the mass spec space through the chromatography, the precursor mass of the peptide, as well as the fragmentation pattern, unlike LBA, where selectivity is only gained through the antibodies and the specificity of those antibodies. Of course, in bottom-up quantification, the process begins at the computer rather than in the lab. The first step is an in silico digestion and bioinformatics analysis that is very important in identifying the signature peptide and checking that there will not be any endogenous contributions in the species of interest. In addition, interruptions further down the development pipeline can be avoided by looking at multiple species across the study design. This is typically done using a suite of software such as XBASI, UniProt, and BLAST. And then once a candidate signature peptide is identified, we then confirm it using high-resolution mass spec experiments following a standard digestion. The high-res mass spec experiments typically use non-specific analysis to identify high-abundance peptides that are unique for that protein or analyte of interest. Once the confirmation is completed using high-res mass spec, then we move on to developing the entire assay, with the most critical piece being the digestion of the protein. Following digestion, this can involve solid phase extraction cleanup, as well as HPLC optimization, as well as detection by the mass spec. The most critical piece in bottom-up analysis and digestion is the digestion itself. This has multiple steps in it. The sample will have the folded protein in it. This needs to be unfolded in order to open up the sites for tryptic or proteolytic digestion. At the same time, this can be done with a reducing agent in order to break any disulfide bonds present in the sample, which produces free thiols. Those free thiols are incredibly reactive, and in order to prevent them wreaking havoc on the sample and to help minimize cross-reactivity, we alkylate it using an alkylating reagent such as iodoacetamide. Following this, we do the digestion with various proteolytic enzymes. The most common that we use is trypsin. However, we have experience using glu-C as well as protein kinase K. We quench that reaction so that all of the samples are digesting at the same rate and for the same period of time. This then moves into a post-digestion cleanup, such as a solid phase extraction, or could be put directly onto the high-performance liquid chromatograph. This is all then detected using the mass spec. Now that we've discussed the overall development process, I'd like to share a specific use case where mass spectrometry may outperform ligand binding assays, and that's specifically in gene therapy. A gene therapy is any one where genetic material is provided to the patient that ultimately is translated into a protein for the function of intent. One challenge in this is being able to monitor both the functional protein as well as the genetic material used as the drug. The functional protein is often very similar to any native endogenous proteins that may be misbehaving or poorly expressed. Often these can be a single amino acid difference. The advantage of mass spec is that we can choose our signature peptide to incorporate that difference and allow us to gain the selectivity that is needed to determine the difference between the native protein and the expressed functional protein. Ligand binding assays may be very hard pressed to achieve the same selectivity when both the native and expressed proteins are present. And this comes all the way back to that bioinformatics start, where you analyze it across species so there's no interruption moving from animal models to humans for the development of the therapeutic. As this slide is showing, the native and expressed may be extremely similar proteins that upon digestion produce unique signature peptides. Even if you can't gain the selectivity through the chromatography, as in this example, the mass spectrum and particularly the fragmentation spectrum will provide you that selectivity to allow you to quantify the functional protein in the presence of the endogenous matrix. We performed a feasibility assessment here at the Colorado Springs site looking at the response of non-human primate and human signature peptides produced from a mixing of human and NHP peptide curves, mixing the two plasmas of each of these. We were looking specifically at phenylalanine hydroxylase. This is an enzyme that converts phenylalanine to tyrosine, and we chose this one in particular because the human and NHP versions are incredibly similar with almost 90% identical FASTA sequences. By selecting a signature peptide that is different, and in this case it was different by only a single amino acid, we were able to quantify the mixed plasmas to identify which one had more human and which one had more NHP contributions. This feasibility assessment could then be applied to a functional protein that is used in a human matrix in which only a single peptide may be different between the two proteins. So how long does it take to develop an LC-MS assay? Well, the in silico screening and high-res mass spec confirmation will take between 1 and 2 days. From there, when we identify our signature peptide that we want to use, we then go and synthesize an internal standard that we can then use to correct for any sample-to-sample variability. The synthesis of that peptide then takes about 2 to 3 weeks. Once we get that in-house, the entire development program will take between 20 and 10 days, which is then followed by a method validation period that is fairly rapid. From there, we can begin sample analysis and within 15 days of receiving the sample, have quality control data available for you. Quality assurance data usually takes a little bit longer. So one advantage of the LC-MS is that it is highly specific. However, in a direct digest, you still have a large amount of biological material available that can lead to complications or interferences that prevents you from gaining the specificity required or the LOQ that you desire. So why don't we incorporate capturing the protein of interest? So then you can use a non-specific antibody that binds perhaps both the native and therapeutic peptides. There are a lot of different names out there for this type of a process where you begin incorporating LBA elements into the LC-MS workflow. We preferred the hybrid LC-MS name. However, it's also been called immunocapture, immunoprecipitation, or immunoaffinity. The main difference between an immunocapture LCMS or hybrid LCMS approach as well and direct digestion is that the analyte of interest is first captured by an antibody, which then allows us to wash away any of the exogenous or extra matrix present in that sample, producing a cleaner sample that we can then digest with our proteolytic peptides and then gain detection using the chemical and physical properties of the peptide. This also would allow us to incorporate a significant amount of multiplexing, such as looking at ADCs. While we will not specifically address ADCs in this webinar, you could see where the free drug or unbound warhead could be separated during the wash step of the capture, and then you only require a single aliquot of sample to determine both the bound and free samples. Overall, the capture provides several advantages. One is that it allows for a pre-purification of the protein of interest, which allows you to get rid of a lot of that extra biological material. This also results in a significant concentration of your analyte of interest, which allows you to put more sample on column when you get to the LCMS and allows you to get to a lower LLOQ. Overall, the process is to first capture the analyte of interest with the antibody. You then take that antibody and bind it to a solid support, usually through a streptavidin type linkage, and that can be either on magnetic beads or some kind of a resin. You then wash away any of the extra biological material and then release it from the solid support in order to go on to the digestion. Another advantage of this is that it allows you to reduce the number of reagents used during the digestion step, which can help keep up your HPLC and mass spec so that you can process more samples between primary maintenance time periods. You can also use off-the-shelf generic antibodies such as protein A or G or anti-human FC or anti-human IgG or any other generic antibody that may not be as specific as you need for an LBA analysis, but is commonly available and readily available. The timeline for developing a hybrid LCMS assay is a little bit longer. The in silico screening and high-res mass spec confirmation are still happening, and you still have to wait for that peptide synthesis for creating the internal standard. The method development does increase by a little bit, and that's because you need to optimize now, in addition to everything going on with the digest, the capture, as well as if you need to release the antigen from the antibody to gain that extra little bit of cleanliness when you inject it on the LCMS. We still can deliver sample analysis data within 15 days for being quality controlled, and our method validation should still be fairly straightforward and only require 5 to 10 days. This results in about a 3.5 month time period as opposed to 3 months as with direct digestion. So what technologies can we at Illyri leverage to deliver your data for life? We have a very large suite of LCMS systems using both nominal mass and high resolution mass spectrometers. And importantly, all of these systems are validated to support regulated bioanalysis. Beyond just the technology for detection that we have, we also have a large group of people with experience that we bring to these analyses. One specific piece of equipment I would like to highlight is the EvoSep that we have here at the Colorado Springs site. This is a special solid phase extraction with microflow chromatography all built into one. So you essentially have an all-in-one sample cleanup with microflow chromatograph. If you think of a traditional SPE, you elute the sample into a sample well and then inject a portion of that solution into a nominal flow or high flow, as I like to call it, chromatography system. This you load the entire sample onto the end of a pipette tip with a small amount of C18 phase resin, and then use microflow to improve the separation and sensitivity of the assay. This allows you to increase your signal significantly, both through loading more sample onto column, as well as achieving greater sensitivity through microflow analysis. In addition to the EvoSep, here at the Colorado Springs site, we use magnetic beads for capture rather regularly. Often that comes in the form of streptavidin magnetic beads. The advantage of a magnetic bead over a traditional LBA where you coat the plate with the antibody is that it increases the surface area that allows you to bind your target. We often wash these beads using automated liquid handling systems, including a Hamilton Starlet as well as Hamilton Nimbus systems here at the Colorado Springs site. And we have taken the starlet and by using a biostriptavidin 5-tip, which incorporates a small amount of resin that has striptavidin on it, we can capture, release, wash, and purify the protein all on tip through an entirely automated process. We also have a standard 300 microliter plate washer with a magnet on site as well for washing those magnetic beads. Here at Illyri, we are FDA and EMA compliant. With a global reach, we have sites in Colorado Springs, Salt Lake City, and France, which allows us to support you no matter where your study is taking place. So where does that leave us? Well, there are several considerations for technology selection, especially for where you are in the development pipeline. I'm going to let Shane discuss some of these considerations as well as why you would choose LCMS or LBA.
Great, thank you for that overview, Matt, of LCMS and LCMS hybrid assays. So one key point in the timeline that we've not talked about yet is method validation and sample analysis. For example, this slide shows the typical method validation acceptance criteria and experiments for a protein LCMS assay. For this validation, selectivity, dilution integrity, matrix effect experiments would be performed following a small molecule mass spec experimental design. However, when a hybrid approach is used, the criteria would be expanded to LBA acceptance criteria. Also on this slide are typical acceptance criteria used for protein LCMS sample analysis. These criteria again follow criteria like those of small molecule drugs, but they are expanded when a hybrid method is utilized. As with all method validations and sample analysis, the experiments and acceptance criteria should be predefined and documented in an SOP or method validation protocol, and regardless of the methodology used, it is essential to employ well-characterized, fully validated bioanalytical methods to ensure reliable results. This will allow for critical go, no-go decisions along the drug development pathway. So what approach should you take? LCMS has timing advantages for tox studies with high LLOQs, since you will likely not require a critical reagent for capture. For a PK study with low LLOQ needs, a hybrid approach using a generic antibody can save time and achieve meaningful detection limits. If high specificity, multiplexing, and a wide dynamic range are important, LCMS or hybrid LCMS is an excellent tool to monitor and quantitate multiple analytes in a single analytical run. In the case of ADCs, LCMS can measure conjugated antibody, total antibody, free payload, and conjugated payload using commercially available reagents like anti-MMAE mAbs and anti-human IgG antibodies. While there are certain reasons to use LBA in specific circumstances, the vast majority of analysis could be implemented faster with LCMS. LCMS provides high specificity quantification of the analyte, and by incorporating elements of LBA into hybrid MS methods, the method development timeline could be greatly accelerated to get your data faster. So I hope that today we have explained the timing benefits of using LCMS for protein analysis earlier in the drug development pathway. Ultimately, we at Illyri want to collaborate with you to provide the highest quality data to advance your drug development pathway. And so with that, I would like to thank everybody at Illyri that helped with this presentation. People were involved from both our Colorado, Utah, and France sites, generating both content and research for this presentation. I also like to thank you, the viewer, for registering and taking your time out of your busy day to watch this webinar. And with that, we're really excited to get into the question and answer session. So I'm going to hand it back over to Kimberly. Thank you very much for your time.
Thank you both for that insightful presentation. We now have some time for some questions from the audience. For those of you who have already submitted questions, thank you. And we encourage others to submit questions just by simply clicking on your Q&A box at the bottom of your screens now. Looks like we've already had a couple of questions come in, so I'll go ahead and get started. So first question, how does the sensitivity of this approach compare to a standard ELISA and an MSD-based or gyro-based assay? Matt, would you like to take this one?
Kimberly. Yes. So the sensitivity between ELISA and MSD is actually pretty critical. So while ELISA and MSD assays have a better sensitivity in general due to the nature of the detector, hybrid LCMS can achieve very, very similar sensitivities, often with better selectivity than traditional LBA analysis. The other really important consideration is that LCMS can incorporate an internal standard that will correct for sample-to-sample variability that is not present in LBA analysis. So even if we can't necessarily achieve the same LLOQ on the extreme end, we can often provide more consistent and robust data in a single sample run.
Thank you, Matt. So the next question, do you need to run samples in duplicate if you use LCMS or a hybrid approach? Shane, do you want to provide your thoughts on this?
Yeah, sure. That's a great question, by the way. So yeah, duplicate analysis is typically done for LBA. I think there's a bit of a move maybe away from doing it in duplicate. I think the M10 does allow for that now, but in the case of LCMS, singlet is all you have to do. And a lot of that is just because of the additional specificity and the inclusion of an internal standard, which increases the accuracy, precision of the LCMS assays compared to those of, a traditional LBA. When it comes to hybrid, you also get that additional specificity by the antibody capture. And then again, you're always looking at retention time of the peptide or the protein and the precursor and product mass. So taking all that together, that's pretty much why you run singlet for LCMS as opposed to duplicate in LBA. Great question.
All right, thank you, Shane. Another one that came in, if the supply of generic antibody runs out, how much validation is required to switch to another antibody from a different vendor? Shane, you want to take this one as well?
Sure, yeah. These are always the problem in modern world, right? So typically, if you're running out of reagent or you need to bridge to another reagent, with LCMS you're going to run one accuracy precision run. You're going to run all your QCs at LLOQ, low, medium, high, 6 replicates. You're also going to include some selectivity, specificity blanks, and that should pretty much bridge between the two different critical reagents. Obviously, you could always do more if depending on the type of study. Another thing that some labs consider too is actually pooling some incurred samples and running those with the original critical reagent and then the new critical reagent, and then seeing how those sample results compare between the two different critical reagents. So you think of it kind of like an ISR test or a bridging study or cross-validation between two different assays. So yeah, great question there.
All right, it looks like we got another one here. How do you save time with LCMS if the run times per sample are longer than analyzing an entire plate with LBA? Matt, can you take this one?
So yeah, the nice thing about LBA is you have your single plate, you put it on the plate reader and you can scan across it with UV-Vis very quickly. The clear advantage, and I think we showed this quite a few times with LCMS, is that your development time frame is a lot shorter. And that comes a lot, especially when you're implementing the hybrid methods, that comes from using more generic antibodies that you can just pull off the shelf and should just, quote unquote, work. The other thing that's helpful with LCMS and hybrid LCMS is that you can be species agnostic. So a lot of times when you develop an antibody, you can pull down your drug of interest in this matrix, but then it has cross-reactivity in human or rabbit or NHP if that's the direction you're going, where that in silico analysis can really help with the front loading the LCMS assays and making sure you can use it through your entire development pipeline rather than having to go back and redevelop something.
Thanks, Matt. Next question. If using a pull down as an initial step, why does this approach offer higher selectivity specificity? Matt, you want to take that one as well?
Oh yeah, of course. So the selectivity and specificity in a hybrid LCMS method is really gained in the precursor and product ions of your signature peptide. That's the real power behind mass spec, and it's been true in the small molecule world, and it's very true now. But you also gain some in the retention time when you're running it on the LC. And you also get some from the antibody. It's not necessarily because we don't need the super specific antibody. You don't gain a huge amount of specificity at the antibody location, but you can help get rid of a lot of the other potential interferences so that you get a lot more selectivity and specificity downstream at the detector.
Thanks, Matt. Let's see here. We talked about sensitivity a little bit, but a particular question on what is the typical low limit of quantitation? The CSF peptides that we want to quantitate is very low in concentration.
So we've developed...
Go ahead, Matt.
Yeah, sorry, jumping in right there. We've developed several assays that across our suite. Hybrid LCMS, we've had as high of an LLQ as a nanogram per mil. However, we've also recently been developing more sensitive assays, especially using the microflow analysis down to the picogram per mil range. So I don't know who asked that question in particular, but we'd love to talk to you about your specific case study because a lot of the work we do tends to be very proprietary and specific to the clients. However, we have gotten down into those picogram per mil ranges that are sometimes helpful on these therapeutic peptides.
Thanks, Matt. Let's see here. What MS platforms is Illyri employing the EvoSep1 on for quantitative bioanalysis? And what SPD samples per day is Illyri using?
I'm happy to take that one, and maybe Matt can chime in as well. So
yeah,
as far as the EvoSep1 goes, we have that actually fully validated to run on our top system. So that's a 7600 Xenotop system from SciEx. We're also can run that on 7500. So really any of our systems that are running SciEx OS, we're able to run the EvoSept on. As far as the samples per day, I think 30 and 60 methodologies we run often, but Matt, I think you run some of the whisper.
Yeah.
Well,
the 100 sample per day is our workhorse one that we run very regularly. We can do all the way down to 30 samples per day. We tried getting some of the whisper methods up and running, but those are significantly more challenging. So there's some development on our end that still needs to happen to make those regular. However, we have used them before.
Thank you both. Let's see here. Do you use triple quads or high resolution mass spectrometers for protein quantitation in a regulated environment?
Yeah.
I'll take that one if you want.
Go ahead, Matt.
In the development side, we always start on the high resolution mass spec, and that's in order to really confirm that in silico analysis. I cannot stress how important that is up front to make sure you're looking at something unique. So we always start on the high res mass spec, but the end goal is always to transfer it to a standard triple quad.
All right, let's see here. We'll give a couple of minutes for some other questions to come in. Looks like we have time for one more question. And let's see. What is the standard smaller volume of sample needed for one hybrid LCMS analysis? Who would like to take that one?
I can take that one. So our standard sample volume is 25 microliters of sample. We've done assays as high as 100 microliters if we need to in order to achieve that appropriate capture. However, we default to 25.
Thanks, Matt. Thank you both and everyone who submitted questions. Unfortunately, we are running out of time. If you have additional questions, please feel free to contact us at the email addresses that are provided here on screen. Once again, thank you for joining us today. We hope you found the webinar informative and valuable. A big thank you to our speakers, again, Shane and Matt, for sharing their expertise and insights. We appreciate your time and effort. We also encourage you all to provide feedback on today's session so we can continue to improve our future webinars. Thank you again for your participation and have a great day, everyone.
Thanks, everyone.
Thank you.